Alright, so in this video, we're going to be talking about dominance, specifically codominance, incomplete dominance, and dominant negative. So, somewhat deviating from our regular Mendelian genetics and talking a little bit about some non-Mendelian dominant methods. So, the first one we're going to talk about is codominance. So, in codominance, both alleles are going to be expressed as dominant, but we're going to get an even distribution of phenotypic features. So something you can think of is type AB blood. Essentially, you're crossing an A and a B, and you're getting AB. So we have a codominant cross right here. So this person's already AB, and this person's already AB, the parents. And but this actually shows you that if you cross two AB individuals, you're still a 1/4 chance of getting an A, and there's a 1/4 chance of just getting a B by itself, and a 50% chance of getting an AB, right, with that genotypic ratio. So for codominance, think even distribution of features, but both alleles are going to be dominant. Another good example for codominance would be like a color of a cow, so the spots on a cow or a chicken. So incomplete dominance is going to be more of blending. So think of blending when we're talking about incomplete dominance. So we're blending our dominant and recessive trait, and we're fully expressing our dominant allele. So our dominant allele is not fully expressed. So a classic example is going to be our crossing our red flower and a white flower. This is going to produce rose color flowers. So say our red is R1 and our R2 is white, right? So if we cross a homozygous red and a homozygous white, we're going to get an outcome of all rose colors for our F1 generation. So it is expressing incomplete dominance. So it's not fully red, but it also isn't white. It's intermediate. All right, so our last one we're going to be talking about is dominant negative. So while a protein is produced, still the protein that's produced is going to be harmful to the human body and to the cell. So it's going to be counterproductive essentially. So a good example is osteogenesis imperfecta. This is going to produce very brittle bones in infants and individuals. So infants are going to be born with incredibly weak bones because the protein is non-efficacious and it's incorrectly formed. And this protein is going to be used for bone tissue, specifically the spongy bone. Here is a quick comparison. So we have our normal healthy bone right here, and then we have our osteogenesis imperfecta. So we have a lot of imperfections here, and our spongy bone area is not correctly made, which is going to make these very brittle bones. So it's going to occur in roughly 1 for every 20,000 infants. Some numbers say closer to 1 for every 15,000. And its most lethal form is perinatally lethal osteogenesis imperfecta. And unfortunately, there's a very high chance of death because these bones are so weak and not always viable. But there are different forms of osteogenesis imperfecta. Okay, cool. Hope you enjoyed the video and hope it helped.